Hi, my name is Joanna. Hi, welcome to Reconnection, and my name is Joanna. Imagine waking up every day knowing that even the slightest touch, a brush of clothing, a hug from someone you love, or the act of eating could tear your skin, leaving painful wounds that won't heal easily, causing relentless itching, um, intense pain, and make everyday life a challenge to just to survive. This is the reality for children and adults living with recessive dystrophic epidermolysis and Bullosa, devastating rare genetic disease where the layers of skin can't stay anchored together, so even normal movements can cause blistering and injury for families affected. The emotional, physical, and financial toll is profound. In today's episode of Our Connection, we talk with a scientist who is working to change all that. Professor Mark Lowdell, Chief Scientific Officer and co-founder of INmune Bio and a transitional immunotherapy leader at the University College in London, joins us to talk about a promising new therapy that has shown encouraging results in clinical trials for RDEB. Welcome, Mark, and thank you for being here. Can you explain to our listeners what makes CORDStrom different from other stem cell or mesenchymal mesenchymal stromal cell therapies?
Yes, first of all, thank you very much for inviting me. It's always good to be able to talk about what we're doing. And as you just alluded to, recessive dystrophic epidermolysis bullosa, or RDEB, as it's called, because no one likes saying recessive dystrophic epidermolysis bullosa, is a horrible disease. It manifests itself in the first few weeks of life, and in its severest form, the median survival is less than 25 years. It's a horrible disease, and as you said, what one sees is this skin manifestation where the epidermis doesn't bind adequately to the dermis. So it's like having, as you say, it's like having blisters over your entire body, which never heal, or they only heal briefly. But the bigger problem is that it's a systemic disease, and every organ in the body other than the brain is affected by RDEB. And there are no treatments for the systemic nature of the disease. So there are some very good topical treatments, and some very, very modern topical treatments. There are even there's a gene therapy, and there's a genetically modified skin graft therapy, together with also an emollient type therapy. But what we were interested in was helping to support a UK trial in pediatric and children with RDEB, following on from some academic work that had been done by colleagues of mine in London, some at King's College London treating adults, and others at Great Ormond Street and University College London, where I work, treating children. And they had some great results in early trials, and they came to us. They wanted to do a much larger trial. And as you said, these mesenchymal stromal cells, sometimes called mesenchymal stem cells, are very challenging. Thousands of trials have been done. Most of them haven't worked. They've all shown them to be safe with some hints of efficacy. And what we wanted to do back in as early as 2014 was try to solve some of those problems. And mesenchymal stromal cells are cells which exist in all of us. They're naturally occurring. They arise in the bone marrow from hematopoietic stem cells, and they travel to sites of damage in the body. They help wound healing. They go into cancer, into tumors, and address the inflammation in tumors, and they're performing tissue repair functions as well. But you can get mesenchymal stromal cells from fat, you can get them from bone marrow, you can get them from umbilical cord tissue, as we do, even from dental pulp. When you look for them, you can find them. The challenge is those cells are all different depending upon the site they come from, but more so, they're different between individuals, even different in the same individual as you age. And so all of these trials that have been done around the world, we'll say very few of them successfully. I think the problem has been, and I think the field now agrees from some of the papers that are coming out, that it's due to the heterogeneity of the material that's been used in these trials. People haven't appreciated quite how variable the product is. If you imagine having a conventional drug like aspirin, every time you go and open a Tylenol bottle, or it's exactly the same drug. That's not the case with most cellular medicines, and certainly when you get into these large-scale medicines like MSC medicines, you're having to use multiple donors to make it. You make one batch from one donor, and then you quite often have to make another batch from a completely different donor. And what we did academically back in 2014, I probably one of the best ideas I've ever had. I thought, well, why don't we combine donors? So we went to the regulatory agency in the UK and said, we'd like to make an MSC product from around 7 different donors. And Lane Godfrey at the UK medicines regulator said, why would you do that? And I said, well, there's heterogeneity between donors. And if we combine 7 donors, you've got a lot of heterogeneity. But the next time we combine 7 more donors, you'll have the same degree of heterogeneity. So although it's not a homogeneous product, it has the same degree of heterogeneity. And she thought that was quite a clever idea and allowed us to do it. And we did an academic trial in the UK, not in Epidermolysis bullosa. And we proved, even in a very small academic manufacturing facility, which I set up 15 years earlier, you could make billions of these cells because it's a much more effective and efficient way of growing mesenchymal stromal cells. And the batch-to-batch variation is absolutely consistent. So when you run out of one batch and you make the next, it's exactly the same as the batch before. And that allowed us in 2019, when our colleagues at Great Ormond Street, led by Anna Martinez, who's the lead pediatric clinician for EB and RDEB in the UK, when Anna wanted to do a trial which was across all of the children in the UK that were eligible, she came to us and said, could we supply the cells? And so we, in order to do that, we spoke to the UK funding agency that funds academic clinical trials in the health service in the UK. And their proviso was that the drug had to be made by a company and not academically. Fortunately, I had co-founded INmune Bio a few years earlier, and the board were happy to take on CORDStrom to support this trial, and that was where it came from. And what came out of it, as you've said, were some really, really encouraging results. And so we have been, we felt, morally obliged to develop this as a licensed medicine, so that all children can be that require the drug can receive it. But what makes me excited is that because we've solved this manufacturing problem for MSCs, other indications of mesenchymal stromal cells can be addressed. And so we're looking at moving outside of RDEB into a number of different diseases. But yes, that's why CORDStrom is different, and it's why we think it's an appropriate way of moving forward in this horrible disease of RDEB.
So are there adults living with this too, or is it how come they tested it with children first?
Yeah, so epidermolysis bullosa is a whole basket of diseases. Probably almost the worst form of it is this recessive dystrophic RDEB, but there are other manifestations. Patients with RDEB, even with RDEB, you get intermediate disease and severe disease, and the severe disease patients tend to die in their 20s and 30s, but the intermediate disease patients can run on until into their 50s, and certainly conventional EB, many patients live admittedly a very difficult life, but they live a full lifespan. So yes, there are adults that need these treatments, and indeed some of the patients in the Mission EB trial that you were talking about at Great Ormond Street and Birmingham Children's Hospital, some of those have already now progressed to beyond the age of 18, so they're no longer pediatric patients, and we have to find a way of managing access to the drug while we're developing it so that they can still be treated.
Right. Yeah, this is the first drug I've ever seen that starts with children, and so it's
yes, the youngest child in the trial was 6 months. We could probably treat children younger than that, but there are certain constraints around clinical trials that treating a child under 6 months would have been quite challenging. So the trial was designed for 6 months onwards.
Yeah. So how does CORDStrom actually work at a biological level?
That's a brilliant question. So one of the other challenges around mesenchymal stromal cells or mesenchymal stem cells treatments has been working out how they work, because when we look at our MSC product, when you grow them, the cells manufacture an awful lot of cytokines. These are the chemical messengers that the body uses between cells to affect an outcome. And MSCs secrete 40, 50 different cytokines depending upon how you stimulate them and whose MSCs you look at. But we had a pretty good idea of some of the potential ways in which MSCs might work. We know they're very good at wound healing, and in fact we've got in laboratory assays where we grow skin cells in a dish and we scratch them and we see how long it takes for those cells to grow back together to heal that wound. And we know that if we take supernatants from CORDStrom, or indeed other MSCs, but CORDStrom, you rapidly increase that because they secrete growth factors. Vascular endothelial growth factor, fibroblast growth factor, many others. And so these growth factors are very important in helping the wounds heal. But as I've said, this is a systemic problem, and a lot of it is driven by itch. The children get very, very bad surface itch and they scratch, and so that exacerbates the wounds. But the wounds themselves, when made, they respond by secreting their own chemical messengers, cytokines, which activates a very complex pathology within the skin. And it's centered around cells which are called tissue macrophages. And macrophages we have in our body to fight infection, and they do other things, but they're largely there to fight infection. They're part of the innate immune response. And there are several different types of macrophages, but one type that's very important causes inflammation. It's there to inflame a site in order for an immune system to get there and fight whatever's going on in that inflammation. You know, we've evolved to deal with infection. And so mostly when you have a skin damage, it's because you've torn the skin traumatically, and therefore you're likely to get infection. So these cells go there. They promote inflammation to get more cells to go there to fight infection. Now, of course, in EB, these RDEB, these children haven't got infection. They've got a traumatic wound, but it's not an infected wound. So that inflammation is becomes a pathology. So what you really want to do is take those inflammatory tissue macrophages and turn them into one of their sister macrophages, which don't cause inflammation, and they promote wound healing. And so the inflammatory macrophages are called M1, and the wound healing macrophages are called M2. And what we have demonstrated both in the laboratory and in the patients is that when you give CORDStrom, you take those M1 cells and you turn them into M2 cells. And that mechanism is well understood scientifically by many other people, and we've now demonstrated that that's one of the principal mechanisms of CORDStrom. So first of all, it reduces inflammation, switches off these M1 phenotype cells, turns them into M2, it promotes wound healing, and then they secrete all of these growth factors that help the wound heal. And that effect isn't just in the skin, it's all the way through the esophagus. And one of the great things that came out of this trial that Ana Martinez set up was that these patients report an improvement in overall pain. If you look at their responses in terms of their ability to eat, it improves. Their mouth pain gets better because this pathology in the mouth is reduced. And so, and if you look at their responses, they get better at going to school, they sleep better, they have better interaction with their siblings in leisure time and all those sort of things. So the general well-being indices go up remarkably in these children after treatment. And I think that the sort of most telling thing about the trial, and I say this wasn't our trial, it was Ana Martinez's trial, but when they interviewed 13 of the parents and children afterwards, and this trial is what's called double blind, so neither the parents nor the child nor the clinicians knew whether the children were being treated with drug or placebo, so it was placebo controlled. And it was a crossover. So half the children got treated with drug first and half got treated with placebo. And then 9 months later, those who'd been treated with placebo got drug and vice versa. But they didn't know whether they were in the placebo first or drug first arm. But before this was all unblinded, the parents and children were asked which they felt they'd got first. Did they feel they'd got placebo first or drug first? And every one of them identified when they'd got drug. They said that they knew they'd had drug because they could sleep better or they could eat better or their itch was less. And I think that was probably the most powerful outcome from my perspective. I'm a pathologist. I like looking at numbers and, what does this blood level change? But actually hearing that these children were able and parents were able to know to determine whether they'd had drug or whether they'd had placebo was really a clear indication that this drug was having an effect.
Mm-hmm. The company plans to submit a BLA to the FDA this year, and similar application to the UK and EU regulators. Can you talk about what the process looks like and what leads you to feel confident about pursuing it now?
Yeah, that's another great question. Getting a drug through to licensing is a whole world different to getting a drug through to clinical trial. And I've done probably 50 clinical trials of different cell and gene therapies throughout my career. And only one other have I taken through to, actually two, taken through to marketing authorization and a BLA. So as you said, in the US, the application is for a biological license application, a BLA. And we will ideally, if all goes to plan, file our BLA around this time next year. The MAA, the marketing authorization, is the same thing in Europe and the UK. And we will be submitting that to the UK regulator first off in about July next year. And of course, when you're going from a clinical trial only treating 30 children into a licensed medicine where you're going to be treating every patient that is available and for forever, not just for a short period of time. The regulatory hurdles are much higher. So the regulator needs to know that you can consistently make the drug. You're not just going to make 10 batches for a clinical trial. You're going to make thousands of batches potentially for decades. And that the facility in which you make this is able to deal with that scale. So you're talking about a scale difference as well. So we've had to move from an academic facility into a facility with a license to make a drug for sale. But also there are things about supply chain. They're really boring things, but really, really important when it comes to a license. And of course, at the end of the day, you've got to be able to persuade the regulator that those clinical trial data are good enough to warrant a license of a drug that people or insurers or whatever will be asked to pay for. And so there's an awful lot of data to be collated into a very large file. And we're working with an expert company called TMC in the UK, TMC Pharma, which specializes in rare disease trials. And I've worked with them off and on for many years. And they have a team of experts available that are helping us get through this process. So yes, we will be submitting to the UK in the middle of next year, followed by Europe, and then following a little bit more work, we will be submitting to the US in December.
Mm-hmm. So how is price decided on the drugs?
So that's also a challenge. Part of it is the price has to be what we can afford to make the drug and supply it for. And then that goes off to either health technology assessors or in Europe or NICE in the UK, National Institute of Clinical Excellence, and they decide whether the cost of your drug has appropriate cost-benefit ratio for the outcome. And it's built on a thing called QALYS, quality of life-adjusted years, and how much will it cost and how many years of benefit will it provide. And we have worked very, very carefully to get our manufacturing costs down to a price where we believe we can meet, at least undercut or match the costs of existing drugs that are used to treat the topical effects of the topical pathologies of RDEB, and if we can add to that in terms of providing a systemic benefit, we hope that that's something that health technology assessors in Europe and the US and NICE in the UK will accept. So yes, a big part of the clinical trial was gathering those quality data to present to regulatory agencies and to funding agencies.
Mm-hmm. So CORDStrom has both rare pediatric disease and orphan drug designations. How do those help in advancing the therapy through regulatory pathways?
So every regulatory agency in the world has a sort of fast track process for rare diseases, and they also give you easier access to regulators for advice during your MAA and BLA. For rare pediatric disease designation, there are other benefits like a lack of fees for submitting your documents. So yes, it's all about ease of interaction with regulatory agencies to get their advice. One of the most expensive things about drug development is having a brilliant idea, getting all your clinical data, putting it all together, submitting it to the agency, and they go, actually, no, what we really needed was this. And I didn't think you would need that. And then you've got to go back and do it. And so these designations allow us to have interactions with the regulatory agencies. We've already had one with the FDA earlier this year. I have had some casual ones with the MHRA, and we are putting together a big submission at the moment to the MHRA for advice. So yes, it's all about access to regulatory agencies so that you don't waste time.
So RDEB is a devastating disease with a few systemic treatment options. If CORDStrom reaches regulatory approval, how could it change the standard of care?
Well, I think what we would look to do is the same as we saw in the clinical trial. that we would. The moment the clinical trial only treated the children, only got 2 doses of CORDStrom. It was an early efficacy trial. What we will do, we're submitting for conditional approval, and then during having been awarded that, we will do an extension trial where the children will be treated 2 doses for 2 weeks every 4 months, so they'll get 6 doses a year. Now, we believe that will end up not only suppressing this itch response and improving their quality of life by improving their esophageal pain, their mouth pain, their joint pain, all of these things we saw in the clinical trial. And as we've seen in the clinical trial, better attendance at school, better sleep, all of these dreadful coincidences to the skin problem. But also we expect, because of this switch, as I said earlier, from this M1 macrophage to M2 macrophage, we expect to see enhanced wound healing, so better management of their skin wounds as well, which, in conjunction with conventional therapies for skin wounds, could have a dramatic reduction in the amount of hospital visits these children have to make for dressings and for management of the skin infections they get. So we hope that it will be a considerable improvement. And after the trial was published, completely without our knowledge, one of the children, and indeed one of the clinicians, was interviewed on the BBC in the breakfast show, talking about their experience. And I think if you look at that, it's on YouTube, you can see the dramatic effect it had for that child in terms of completely changing his ability to interact with siblings and go to school. So yes, we hope to see those sort of findings enhanced even with the repetitive treatment.
Now, the CORDStrom platform itself is a disease agnostic. What other conditions might this platform eventually be optimized to treat, and what research already is underway?
So we were also delighted to supply the cells into an academic trial in Paris. A good friend of mine, who is a rheumatologist at Hôpital Saint-Louis in Paris, wanted to do a trial in patients with this systemic lupus, the so-called adult butterfly rash disease. And we supplied the cells into Dominique Farge at Hôpital Saint-Louis, and that paper is published in Lancet Rheumatology, where we showed significant clinical benefit to ladies with systemic lupus. Others using similar products to CORDStrom have shown benefit in osteoarthritis. Indeed, there's a licensed medicine in South Korea made from MSCs, albeit not pooled, in osteoarthritis. So we have a lot of interest in osteoarthritis. The original creation of CORDStrom back in 2014 was to be genetically modified to carry a molecule that kills cancer cells. So the immune system, when it's working properly in cancer, kills cancer cells by a molecule called TRAIL, TRAIL. And a colleague of mine at UCL, Sam Janes, had the idea of putting TRAIL into MSCs because, as I said very early on this evening, that MSCs track into tumours as part of the innate repair mechanism. And so he demonstrated in a mouse model of lung cancer that these cells carrying TRAIL would track into the tumour and kill the tumour. And so he wanted to do it clinically, and we invented CORDStrom for that very purpose. And that trial is now, the paper's in press. So we've demonstrated we can deliver cancer-killing molecules on CORDStrom into tumours. And so we have an interest in moving that forward as well in the cancer space. And we have another study I've been collaborating with colleagues at the Institute of Neurology at UCL, where we've used the anti-inflammatory effects of CORDStrom as an intranasal delivery to deal with neuronal inflammation in a piglet model. And we're putting together a grant application next year to support a clinical trial with our colleagues at UCL and the Institute of Neurology. So because we pool donors, so the CORDStrom is made from mesenchymal stromal cells isolated from umbilical cords. And these cords are cords that have been donated to the UK National Cord Blood Bank, and they're not needed for cord blood. And so we take these cords and we can grow those out as individual mesenchymal stromal cell products and look at the characteristics. And as I've said, every donor has different characteristics. And so we select 4 donors with the characteristics we want to target a particular disease. And then that's what we pool and grow up. So CORDStrom can be targeted to osteoarthritis by getting cells that have the right cytokine response for osteoarthritis. We can do the same for systemic lupus. We can do the same for a horrible disease called Sjogren's. So at the moment we're focusing on rare diseases, and we will tune our different CORDStrom batches as different drugs to target different diseases as a true platform. And I think that's what really excites me. We have this manufacturing platform that is very reproducible, and we can tune it to target different diseases.
Yeah, I noticed in the article I was sent that you had something for Alzheimer's that you were looking into also.
Yeah, so this is Immune Bio has been includes we hold 3 drugs, 2 of which are drugs that I invented, and the third we bought in called Expro, which is a unique drug. It was invented in the US by a company called Xencor, and we bought it into the company in 2017. And we've just completed a phase 2 trial in Alzheimer's. And what's become apparent only in the last few years is that Alzheimer's is a disease of neuroinflammation. And one of these chemical messengers that I was talking about earlier on that drives that neuroinflammation is a molecule called tumor necrosis factor. It's involved in all sorts of other inflammations. In fact, severe rheumatoid arthritis is driven by tumor necrosis factor. Now, TNF has 2 different forms. It's made in a cell or in cells. It sits on the surface of the cell as membrane-bound TNF. And as immunologists, we tend to think of that as good TNF. It provokes the immune response. It does really important things in helping you make an immune response to viruses and cancers. But in times of inflammation, that membrane-bound TNF gets shed and becomes soluble TNF, and that causes a lot of inflammation. And in some diseases, like Alzheimer's, it causes a lot of pathology. And so it would be great to be able to block that soluble TNF. However, all of the drugs that are anti-TNFs at the moment block both soluble TNF and transmembrane TNF, and so they have the toxicities of being immunosuppressive. Expro is unique in that it only blocks soluble TNF, and it stops it binding to the receptors in the brain that cause some of the brain inflammation. And we were able to show in the clinical trial, which completed and was published, the results were made public in July last year, showing that patients who had 2 biomarkers of neuroinflammation, so a lot of brain inflammation, that Expro resolved that and led to improved remyelination and reduction in the brain pathology. So we're very excited by that. We're now looking at ways to move that forward, and the data have been sent to the FDA for discussion probably in January or early February next year.
Wow. So, Mark, thank you so much for being here today and for sharing your insights, your experiences, and most importantly, your passion for turning science into a real hope for patients and families. Your work reminds us that a connection between researchers, caregivers, patients, and community is powerful. To everyone listening, if you found this conversation, this episode, please, if you found value in this episode, please subscribe to or share and leave a comment. Rare Connection is built on community, and your engagement helps us reach more people who could benefit from these conversations. We'd also love for you to join our Rare Connection Facebook group, a safe, supportive space where listeners, caregivers, patients, and advocates share stories, resources, and encouragement. And for anyone following multiple diets or navigating complex nutritional paths, don't forget, you might want to check out the Adaptive Kitchen Facebook group, a place for people managing multiple dietary needs and sharing practical tips and recipes. Links to both groups will be in the show notes. Before we go, a reminder, if you or someone you know is struggling emotionally or experiencing thoughts of suicide, help is available 24/7. In the United States, you can call, you can't, United States and Canada, you can call or text 988 to reach the Suicide and Prevention Lifeline. And you can also text 741 home to 741-741. I believe that's also in the UK. To connect to the crisis text line for a free text, for a free 24/7 text support service. You are not alone. And reaching out for help is a strong and important first step. Thank you again for listening, and take care of yourself and take care of each other. And we'll see you next time on Rare Connection. Thanks.